program for you in the afternoon, not to miss, and party in the evening. So again, drinking, networking. And I'm very happy that we have now our last speaker, who is our local organizer. We work together in a way already since a couple of years. Means actually since Anna is part of my group, we are publishing together, but I haven't met him in person before. So I'm more than happy that I had the chance to organize this together with him here and meet him here. And I now see you also in person, Basil Gurmach, his co-worker, and you are presenting together, or he is presenting? Ah, okay, he is presenting. Okay, I thought you are presenting.
Yes, corresponding.
Yes, you are corresponding. Okay. Okay. So then you will introduce him. I will just say a few more words, not much. So this is the last. Talk of the scientific part, and from the title, which is now gone from the screen, you may already get an idea. Thank you. You may already get an idea that it brings really together, hopefully, these two fields, nanotechnology, now here represented again by first the fullerenes, then also the viruses, also nanoparticles, and on the other hand, computer science, maybe AI, represented then by an in silico inside. So I welcome now Professor Yuri Prilutsky to present his co-worker. Thank you.
Summarize it. The next speaker is my doctorand, current doctorand in the field of theoretical biophysics, Dr. Vasil Gurmach. His lecture is devoted to the possibility to create nanomaterials based on C60 fullerenes against pandemic COVID-19, SARS-CoV-2. Okay?
Hello, dear colleagues, students. Here I want to present the project Yuri Prilutsky group has been working on for some time. The work was done at the Russian National University of Kyiv, and as you can see, that is carbon 60 fullerene against SARS-CoV-2 coronavirus and in silico inside. The coronavirus is actually the virus that infected mostly vertebrate. Here you can see some insights in the structure of the virus. And actually, as you know, currently coronavirus is widespread all over the globe. So far, about 2 million cases have been performed, including as I remember, about 5 million test cases. So, as I mentioned here, you can see the structure. So, about the size of coronavirus, the size of the size of which we are in diameter can vary from 60 to 140 nanometers, and as all the other coronaviruses, SARS-CoV-2 contains the main proteins like spike protein, envelope, membrane, and nuclear acid protein. Spike protein is responsible for binding of coronavirus to the host cell, together with envelope and membrane proteins. They create envelope of coronavirus, and of course nucleocapsid protein called RNA molecule of coronavirus inside the virus. So, and besides those proteins, actually, there are a lot of other proteins which related with the work of coronavirus. You can see them in the bottom part of the slide that is different structure proteins, 3C light protease, and actually so on. So, as you actually understand from the name of presentation, all the modeling was performed with carbon-60 fullerene. Here you can see the structure of the molecule, and why we actually did it, because currently there are a lot of investigations which in which people try to inhibit coronaviruses with different classes of chemical compounds, but there is no of such investigation with any nanomaterials. So, and actually, we think that it could be some alternative resource to fight coronavirus in different stages of infection. And another reason we did it because, according to literature data, carbon-60 fullerene has antioxidant, antiviral, and some membranotropic activities. And the next stage we have done, it was that we should select the target of as target actually for the carbon-60 fullerene, and for that we checked a really huge pool of proteins. There was more than 15 proteins we checked, and amid various viral proteins, we have selected two which is irreplaceable for virus life cycle. The first one, that is 3C light protease, and this protease has a critical role for replication and expression of viral genome. And as you can see, it has unique catalytic binding pocket. Here you can see the full structure of monomer, and in the catalytic binding pocket, it has the catalytic DNA, which formed with histidine 41 and cysteine 145. And actually, this interaction is critical for its functionality, and by shielding it, we can actually probe the chain of functionality of this protease. And the other target we selected, that is RNA-dependent RNA polymerase. So, but it's not it's only because we tried to investigate it in conflict with non-structured protein A. You can see it here in violet, and non-structured protein cell. Because actually, only in such association it is possible to activate the replication of long RNA molecule, of the long RNA virus molecule. So here you can see the RNA molecule surface of RNA-dependent RNA polymerase, two non-structured proteins, and actually the RNA synthesis chain. And then based on that, actually, I want to mention to you the main idea of our project. So, according to my idea, it doesn't mean we want, for example, fully inhibit our protein by fullerene. We wanted to create some calculation, some starting point for other kind of calculations. For example, you can take these both proteins, take our models, 1, 2, and try to check this model by using any other kind of chemical compound, any other class of chemical compound. And actually, as we expected, in the output you will get the inhibition of these two proteins, and as a result they would not be able to perform the initial functions. So after we select the protein target, in the first stage, we should determine the possible binding pockets for carbon-60 fullerene. Here you see the small flowchart of this stage of calculation. So in the first stage, the literature and structure data analysis helping time. After that, we analyze all the binding pockets we can find in the literature data, and selected PDB structure from protein database to carry out the calculation, and actually analyze the surface as well. And the next stage was the surface analysis for selected PDB structures for C60 protease we selected. This protease, it is a different protein data bank, and for RNA-dependent RNA polymerase, it was that. And as a result, for C60 protease, we selected one possible mechanism of inhibition, and for RNA-dependent RNA polymerase, we selected two possible three-body pocket and two possible mechanisms of inhibition by carbon-60 fullerene. Okay. Here you can see the full structure of C60 protease, catalytic decay, then increased size of catalytic decay. I can explain why it's necessary before that. And the surface analysis. We actually did the surface analysis of full structure. The surface analysis of full structure, but the only possible way to blind this fullerene was this catalytic body pocket because of this volume. As you can see, the volume is really huge, about 900 cubic angstroms. So that is the first site we selected actually, and the second protein we analyzed, it was RNA-dependent RNA polymerase. So first binding pocket selected, it was is located inside the RNA synthesis shell. Why actually we did this? Because there we find cavity where RNA synthesized RNA binds. Binds pretty well, and this pocket contains such amino acids like arginine, lysine, tyrosine, which could actually create a stacking interaction with carbon-60 fullerene. So, and the idea was the idea we have here is simple, because if here we have carbon-60 fullerene, there will not just enough place for RNA molecule and RNA synthesis will be stopped. So here is simple idea. The other binding pocket we checked was in the binding interface between a non-structured protein and RNA-dependent RNA polymerase. And actually, why we selected this pocket? Because it is contains such amino acids like tryptophan, tyrosine, thymine, two lysines from different sites of protein. We actually hope that those vinyls would clump carbon-60 fullerene inside the binding pocket, but the interaction with carbon-60 fullerene after molecular docking was questionable. It was questionable, and why? Because actually the conformation of this pocket is really flat. So it means that a complex, there is a possibility complex will not be stable. Yeah. And the pocket number 3 we selected is here in the binding interface between non-structured protein RNA and RNA-dependent RNA polymerase as well. But actually, what is interesting about that pocket that this pocket doesn't contain any amino acid which can create aromatic cation or any other stacking interaction with carbon-60 fullerene. What you can see here, it is only some amino acid which could create steric interaction with carbon-60 fullerene, and only steric interaction with carbon-60 fullerene. But why did it select this pocket? Because of its dips. So that pocket is really deep, and we hope that, for example, during this simulation or something like this, carbon-60 fullerene would immerse inside the binding pocket, and because of that, the complex will be stable. Okay, and the last part we analyzed here, it was the interaction between RNA-dependent RNA polymerase and complex of non-structured protein A in violet and non-structured protein 7. But actually, what we have found here is that binding interface is really flat and it doesn't contain good for us amino acids. We could create some stacking interaction. And to be honest here, it would be even difficult to interact with any ligand here. So this is why, and especially with carbon-60, this is why this binding pocket was rejected. So, and small conclusion about this stage is that actually we selected one of three possible binding pockets for carbon-60 fullerene incomplete for carbon-60 fullerene with C60 and RNA-dependent RNA polymerase respectively. And actually, if anybody has a question about this stage, because presentation a bit long, you can ask. Yeah. To analyze the structure, it was for that is a programs like Cover, Pymol. You can use Cover, Pymol, Chimera. But if you don't know Chimera, please download the new one because there is two versions and they proposed both of them. But the newest is really good and old one is bad. That's really bad. Okay. So
the next step after this in silico, what you will do next?
No, the next step actually a lot of in silico more, and I hope the some biology testing. You know.
No, I know, but which or you
I can answer this because we didn't consider it yet. So, sorry.
or you need the protein of the virus, or what do you need
For testing?
For verification?
Yeah, we need protein, or yeah, I said we need the protein, some protein inside. I hope we will do it in the future, but no, unfortunately not. Yeah. Okay, any other questions? Okay, so I can conclude. So the next stage we did here, it was the construction of carbon-60 fullerene and our proteins system. So for that, here you can see some small slow chart of this stage. First stage was intimate all-routine water molecules and native ligands from our protein structure, then energy minimized our protein structure. Simultaneously with that, the structure of carbon-60 fullerene were retrieved from Campbell database, and the structure of this of carbon-60 fullerene was generated. Then, by using flow transfer tool, molecular dating calculation was performed. To each calculation, 10 test complexes based on Q-scoring function were selected for actually visual analysis, and after visual analysis to each calculation, only one complex was selected. Okay, so. Here on the slide you can see the presented full binding model of one monomer of C60 protease and carbon-60 fullerene in a catalytic binding pocket, and increased size of binding pocket and binding with fullerene. And what is interesting here is that carbon-60 fullerene completely shields the catalytic DA, and as you can see, it is even hard to see histidine-45 behind the carbon-60 fullerene. It means that they have good direct stacking interaction. Also, carbon-60 fullerene surrounded by different amino acids like cysteine, 104, no, cysteine, methionine, 160, asparagine, arginine, and carbon-60 clump by that inside the binding pocket. But the most important thing here is that it shield the catalytic gas, so that is why C60 protease doesn't work. Okay, and the other protein RNA-dependent RNA polymerase, here you see the full structure with carbon-60 fullerene bind inside the RNA synthesis channel, and actually, as you can see, it fills a lot of space inside this channel. It fills a lot of space and bigger picture here, and here it makes a carbon-P interaction with this lysine, this arginine, and possibly just taking with this tyrosine and a lot of other steric interactions actually with amino acids environment. And other binding model that is simultaneous binding of carbon-60 fullerene with binding pocket number 2 and binding pocket number 3. And what we do here, what we have here is this carbon-60 fullerene clump by these lysines makes steric interaction with this histidine, tyrosine, just steric with this phenyl, and some steric interaction, not steric, stacking interaction with this tryptophan. And in other binding pocket, as I said, it's surrounded by amino acid environment which can make steric interaction with one only. Okay. And conclusion from this slide, actually, is really the main conclusion is that carbon-60 fullerene potentially can create complexes with C60 protease and RNA-dependent RNA polymerase. And any questions about that slide? Because we have few more. Okay, if you don't have any question, let's go.
Which is the level of specificity these two proteins, but there are 10,000 proteins.
We tried to answer that question further in the presentation because unfortunately by molecular docking simulation, I think we can't answer the question. Of course, we have docking score, but it is a bit abstract thing, you know, like, and I'm not sure we can say that by this we can evaluate the specificity of, you know, like carbon-60 fullerene to those proteins. So, unfortunately, this stage can't answer your question, but we will try to do it in our further stage. Okay. So, and the other stage we did to obtain more accurate result and estimate the stability of training complex, the MD simulation was performed, and actually here you can see the full flow chart of calculation we did. So, all the calculation was carried out in the Gromov tool. And actually, the first stage was generation. We generated the topology of our protein structure by using PDB to GMX function, and by using Swift farm server, we generate the topology file for carbon-60 fullerene. The next stage, we create complex of carbon-60 fullerene, reset-up complex of carbon-60 fullerene and our proteins. Parallel with that, topology file for reset-up complex was generated as well. Then put our system to the cubic box, and the distance between, and to be sure that actually proteins will be able to freely rotate inside the box, the distance between any nearest atom and the edge of the box was 9 angstroms. Then solvate our system and add ions in the cellular concentration to imitate cellular environment. Then run energy minimization with our complex two-stage of equilibration with a stable temperature and stable pressure, and of course MD simulation, full MD simulation. Then analyze our structures, analyze root mean square deviation, root mean square fluctuation, did some visual analysis, something like this. Okay, so here you can see the result of MD simulation result of CC-like proteases, carbon-60 fullerene, and what actually we got there? We got some shifting of carbon-60 fullerene during the MD simulation for about 3 angstrom, and it actually caused some other shifting of amino acid, but they wasn't critical actually for full structure. What was critical here? That carbon-60 fullerene forced out histidine 45 here from its initial position, and because of that, the integrity of catalytic diode was violated. So here you can see cysteine 145 and histidine 41, and as you remember in the some of first slide, I mentioned that actually this H-bond interaction here is really necessary for its functionality. And by carbon-60 fullerene, during this simulation, you violated this integrity. And this is that without any doubt, for us they good because it will not function. Here you can see that element of the trajectories in the red that is protein trajectory, in the red it is carbon-60 fullerene trajectory, and in the green it is protein trajectory. So as you can see, they stable. Carbon-60 fullerene has a bit more stable trajectory compared to protein, but that is actually. And actually, you can see the violated catalytic, broken catalytic here. It is the main result of this stage for CC-like protease. And about indeed of RNA-dependent RNA polymerase for the first pocket, no any significant changes were observed. For pocket number, we have actually the same situation. For Python pocket number 3, everything was interesting because, as we expected, carbon-60 fullerene start to immerse inside Biden pocket, and except some steric interaction with amino acid environment, we start to make steric interaction. Unfortunately, somehow it doesn't clear. So with this tyrosine and something might be phenyl, but I'm sorry, I don't see it here. I don't know why it doesn't clear. Okay, so we have some alveolar pocket 2 and 3 without knowing changes. And some conclusion about this state. And the main conclusion about this is that actually the complexes we built show the stability during full MD simulation, and even though for each MD simulation we had at least 3 iterations. So something. If somebody have any question about this stage.
Yeah. Taking into account the shape of these fullerene balls, they cannot be immobilized very strongly. It will move, and comparing to other inhibitors.
Yes, for sure you are right. I got it. You are right, but but on the one hand, it is good, but on the other hand, because of such kind of shape, it could be easily forced out from the binding pocket during the simulation by some amino acid. And actually, when I simulate carbon-60 fullerene with some other protein, I got such results for a lot of time. So.
Is there any indicator of such strengths of interaction?
There are few types of calculation we can use here, and one of them I will show you in the presentation. I think actually this is the next stage. Okay. Okay, so the next stage I want to present you evaluate more precise the binding between our protein targets and carbon-60 fullerene, related binding between each amino acid and carbon-60 fullerene. We write this stage of calculation with MMPBCA, MMPBCA calculation, and actually here you can see the full flow chart that in the first stage we actually take the full trajectories of MT simulations, then to each of trajectory and each snapshot of each trajectory we calculate potential energy between protein and carbon-60 fullerene in vacuum, then calculate polar solidation energy between them, then calculate non-polar solidation energy, and based on those energy, we calculated binding energy, Gibbs energy. And here in the table you can see the result. And what is interesting about those results that, oh, I'm sorry. That G binding for carbon-60 fullerene in conflict with 3-C-like protease and RNA-dependent RNA polymerase. Pocket 3 is more favorable. Pocket 3 more favorable compared to binding with RNA-dependent RNA polymerase with pockets 1 and pocket 2, and except this, we can distinguish the impact of each energy to complex formation. And according to this table, we can say that the advanced energy have the main impact of our interaction, and electrostatic polar and non-polar energy actually they close to null or null or even bigger than null. And actually that means that they have negative impact for complex formation. And in the other stage, we run MMGBCA calculation, but by this calculation, we try to estimate energy between each amino acid of protein and carbon-60 fullerene. So here you can see the results for 3-C-like protease, and as we expected, all aromatic amino acids create good stacking interaction with carbon-60 fullerene. But what was surprising that arginine 40 has unfavorable binding energy with carbon-60 fullerene. You can see it here, and it means what you did mention, that here arginine 40 forced out carbon-60 fullerene from the binding pocket, so the interaction actually is negative for complex formation. And here you can see the same results for the RNA-dependent RNA polymerase binding pocket 1 and binding model 1. I'm sorry, binding model 1. Actually, we have the same good interaction with a huge pool of amino acid which has some aromatic part, and again bad interaction with this arginine, and actually here it this tyrosine. It doesn't work. I'm sorry. And the same results for binding model 2, binding pocket 2 and 3 actually. So, and we don't have there any unfavorable amino acid. Some conclusion about this stage I would not read them because I actually read them during the presentation, this stage of presentation. As a result of this work, we have published publication in Nature Communication. You can see the title here. And the main conclusion, and as for me, the main conclusion of this work is to be fully analyze those two proteins and create some models which could be used as a starting point for any other calculation or work of such type. As they are mentioned in the start of the presentation, for example, you can take any other type of compounds, take our model and just check them via our models. So, and thank you.
Did you have positive controls to compare the affinities and other indicators within?
You mean during the simulation?
These simulations, yes.
Yes, we compare.
Because there are no on the table.
Yeah, we didn't present them, but actually we compare our result with some, let's say, ligands. Other researchers think that they would work against coronavirus because currently actually we don't have such compounds as I know. So, and honestly, the result was pretty the same. They wasn't the same. There was some distance in the bar, but they were close. So
more questions, please, from audience. Please.
Can you please go back to the 30 SARS? For example, in the COVID-3 and RLV, the energy of binding is 150 kilojoule per mole. I know that, for example, the B molecule, the energy of binding, this molecule is 30 kilojoule per mole. But there is a covalent binding. If I correctly understand, there is no covalent binding. How it is possible so high energy?
yes, it is a problem of such kind of methods. Because actually, I'm not sure, that like we can refer fully to this number. These numbers can present just a picture, just a relation part. For example, if I use some other kind of method, for example, you calculated the covalent bond and calculate the energy for this complex, I'm pretty sure the numbers will be other. So this is a problem for those methods. They could say only the relation. They can, unfortunately, they can't say show exact picture.
Maybe it is possible because there is a relation with few amino acids and everything. Small contribute.
That is questionable, honestly. So they can show only the relation sometimes.
The second one,
Actually, could you please be a bit more loud?
A bit loud.
Yes.
can I realize docking in my laptop?
Yes, you can realize, for sure you can realize docking in your laptop. Docking is not a calculation method that costs a lot of resources. If you are talking about some quantum calculation or this simulation, it really needs a lot of, but docking doesn't.
So more questions, please. You are welcome. Don't be shy. Ask. It's a great opportunity for you. And we have plenty of time. 30 minutes, I think. It's really all, yeah? So you already finished your lecture, yeah? Okay.
Yes.
So please, dear students from both nano and computer science. So it looks like we should.
Okay.
Yeah,
No
thank
more
you.
questions. Thank you.
Then I propose, so I'm not sure that our lunch is ready. I should ask Anna, but if it's ready, we can proceed. But if it's not, we should wait, or we can launch some discussion or free time. Up to you. Now I ask